Hello students, Jinalisha welcomes you all on behalf of Saraswat International Academy. Today we will begin with the new chapter Photosynthesis in Higher Plants. So basically we are going to start the new unit called as Plant Physiology. So what is photosynthesis? We can split this word into two terms. Photo means light, synthesis means to produce. So plants, they produce or they synthesize their own food in the presence of the sunlight. That process is called as photosynthesis. So how this photosynthesis process occurs, we will be studying in this particular chapter. So let us begin with the chapter Photosynthesis in Higher Plants. So first of all, the introductory part of the chapter. Why are green plants necessary for living organism? What is the importance of the green plants to living organism? Will life be possible if green plants are not there? So we will see why the green plants are very much necessary for all the living organisms. All animals, including human beings, depends on plants for their food. Each and every living organism inhabiting the earth depends on plants for their food. Green plants in fact have to make or rather synthesize the food they need, and all other organisms depend on them for their needs. So what green plants do? They synthesize the food for themselves as well as the other organisms also depend on them for their needs. That is, green plants, they prepare their food for themselves as well as the other organisms also depend on the green plants for the food. The green plants make or rather synthesize the food they need through photosynthesis and are therefore called as autotrophs. Now the green plants, they synthesize their own food by the process of photosynthesis. How do green plants prepare their food? They prepare their food by the process of photosynthesis, and hence the green plants are known as autotrophs. Auto means self and trophy means nutrition. So those organisms which prepare their own food, they are called as the autotrophs. The autotrophic nutrition is found only in plants, and all other organisms that depend on green plants for food are heterotrophs. Now, they are only the green plants and some few blue-green algae, or you can say certain photosynthetic bacteria, which can prepare their own food. So basically autotrophic nutrition is found only in the plants and few prokaryotic organisms. Otherwise all the other organisms depend on the green plants for their nutrition. So those organisms which depends on green plants for their nutrition are called as heterotrophs. What are they called as? They are called as the heterotrophs. So green plants, all the living organisms including we human beings, we depend on the green plants for our food. The green plants, they synthesize the food for themselves as well as for the other organisms, and they synthesize the food by the process called as photosynthesis. Their mode of nutrition is called as autotrophic mode of nutrition, and the other organisms are dependent on the green plants for their food. So these organisms are called as the heterotrophs. Green plants carry out photosynthesis. It is a physico-chemical process by which they use the light energy to drive the synthesis of the organic compounds. Now who carry out the process of photosynthesis? It is the green plant. Now this photosynthesis is a physico-chemical process कि जहाँ पे they use the light energy and they convert that light energy to chemical energy. That is, using the light energy, they synthesize the organic compound. The photosynthesis is a physico-chemical process where the green plants use the light energy for the synthesis of the organic compound. Ultimately, all living forms on earth depends on sunlight for energy. See, it is the green plants which prepare the food for all the organism. Other organisms are dependent on green plants for their food. Now, green plants कैसे food बनाते हैं? So, the green plants prepare the food using the sunlight as the source of energy. So, we can say that sunlight is the ultimate source of energy for each and every living organism on the earth. The main source of energy for each and every living organism is the sunlight. See, ग्रीन प्लांट्स सनलाइट को यूज़ करके फूड बनाते हैं एंड ये फूड को कौन कंज्यूम करता है? दूसरे ऑर्गेनिजम्स. सो इनडायरेक्टली इट इज़ द सनलाइट विच इज़ द अल्टीमेट सोर्स ऑफ एनर्जी बिकॉज द ग्रीन प्लांट्स कैन नॉट सिंथेसाइज द फूड इन द एब्सेंस ऑफ सनलाइट एंड ग्रीन प्लांट्स जो फूड बनाते हैं उसको कौन कंज्यूम करता है? दूसरे ऑर्गेनिज्म. सो मेन सोर्स ऑफ ऑल द लिविंग ऑर्गेनिज्म, मेन सोर्स of the energy for all the living organisms is the sunlight. The use of energy from sunlight by plant doing photosynthesis is the basis of life on the earth. So what is the basis or how can we say that life is possible on the earth? Because of the use of the sunlight by the green plants. Photosynthesis is important due to two reasons. There are two reasons which makes the photosynthesis an important process. It is the primary source of all food on earth. Whatever food on earth is prepared, उसमें main जो source है food source, so it is basically through the photosynthesis. It is also responsible for the release of oxygen into the atmosphere by green plants. So दूसरा reason क्या है? By the process of photosynthesis, the oxygen will be released into the atmosphere. So these are the two main important reasons for photosynthesis. First, it is the primary source of all the food on the earth. जो main food source है वो कौन है? Green plants. And green plants कैसे बनाते हैं? By the process of photosynthesis. So main source of food for all the living organism is coming through photosynthesis. And during the process of photosynthesis, oxygen is released into the atmosphere. Green plants, they release the oxygen into the atmosphere at the time of photosynthesis. And we know the importance of the oxygen. This oxygen is, the use of this oxygen is done by every living organism in the process of respiration. So, oxygen released by the green plants is used by the living organisms in the process of respiration. Now, some history related to the photosynthesis. So, what information do we have regarding photosynthesis? Some simple experiments you may have done in earlier classes have shown that following are required for photosynthesis to occur. During your lower standards, you might have studied certain experiments, and from that experiments, you might have came to conclusion that there are certain requirements of the photosynthesis. Which are those requirements? First is chlorophyll, which is the green pigment of the leaf. The second one is the light, whose main source is the sun, and the last thing is the carbon dioxide. Chlorophyll, it is a green pigment which help to trap the sunlight. प्लांट सनलाइट को यूज़ करके फूड बनाते हैं, लेकिन ये सनलाइट को एब्सॉर्ब कौन करता है? सो इट इज़ द क्लोरोफिल पिगमेंट, सो विच हेल्प्स इन ट्रैपिंग ऑफ द सनलाइट. Light, which is the main source of light, it is the sunlight. Using the sunlight only, the process of photosynthesis will take place. And carbon dioxide, it is the carbon dioxide which gets converted into the carbohydrate, which is nothing but a glucose. Which carbohydrate will be made? Glucose. And this carbohydrate is nothing but it is the food prepared by the process of photosynthesis. So these are the three main requirements of the photosynthesis. One is the chlorophyll pigment which helps to trap the sunlight. Then light जहाँ पे photosynthetic activity होगी जिसके presence में, and carbon dioxide which is converted into carbohydrate which is the food source. Now, what we have studied in your lower standard. So, first of all, you might have studied the two leaves experiment for the formation of the starch. So, a leaf of a healthy developing plant that is partially covered with black paper, that is variegated leaves, and the other one is exposed to the light. So, two leaves, one with one leaf which is partially covered with the black paper, another one which is exposed to the light. Now, after some time, a test is done with dilute solution of iodine in both the leaves. Now, test is done after few minutes, or you can say after some time. A dilute solution of iodine is used to test the presence of the starch in both the leaves. So, what observation do we get? Starch is formed in a leaf which is open. So, in a open leaf the starch is formed. कैसे हम बोल सकते हैं कि starch बना? So, it becomes blue by iodine. So, जहाँ पे photosynthesis हुआ होगा वहाँ ग्लूकोज बना होगा, वो ग्लूकोज स्टार्च में कन्वर्ट होता है, and that starch can be detected by the presence of the iodine. So, जो open leaf था वहाँ पे जब starch की टेस्ट की गई, then that leaf was giving the blue color which indicate that the presence of the starch, whereas the leaf which is covered partially with black paper did not stain, that is did not showed the presence of the starch. So जो open leaf था जहाँ पे black cover नहीं था वहाँ पे photosynthesis होकर starch बना, and जो covered leaf था black covered जो leaf black paper से covered था, that leaf did not showed the presence of the starch. So what important conclusion can be drawn from it? This experiment shows that the process of photosynthesis occurs in green parts in the presence of sunlight. ये experiment से ये conclusion आता है कि the process of photosynthesis occurs in green parts of the leaves in the presence of the sunlight. A very important conclusion drawn from this experiment is the photosynthesis occurs in the green parts of the leaves in the presence of sunlight. Clear? Now, next experiment is a half leaf experiment. In this experiment, where a part of a leaf is enclosed in a test tube containing some KOH, KOH is potassium hydroxide, soaked cotton. Now, KOH has the property to absorb the carbon dioxide, while the other part is exposed to air. Now, in this experiment, half of the leaf is dipped into the test tube which contains the KOH soaked cotton, and other part of the leaf is exposed to air. Now, what are we checking? We need to check the presence of the starch after some time in two parts of the leaf. So, what is the main thing? We need to check the presence of the starch in two parts of the leaf. That is in the part which is dipped in a test tube where KOH soaked cotton is present, and other part is exposed to atmosphere or to air. Now, what happens? The setup is that placed in the light for some time. It is observed that the exposed part of the leaf tested positive for starch, while the portion that was in the tube tested negative for the starch. So, क्या observation मिला ये test में से कि जो part, that part of the leaf which was dipped in the test tube containing the carbon dioxide soaked KOH soaked cotton, वो पर्टिकुलर पार्ट में स्टार्च प्रेजेंट नहीं है, whereas the exposed part of the leaf showed the presence of the starch. So, from this experiment, it can be concluded that the carbon dioxide is very much essential for the photosynthesis. क्यों KOH वाला जो cotton था और why the leaf which was dipped in the test tube didn't show the presence of the or didn't show the presence of the starch. The reason is वहाँ पे जो भी कार्बन डाइऑक्साइड था उसको एब्सॉर्ब किसने कर लिया? KOH. And in the absence of the carbon dioxide, the carbohydrate cannot be formed. So this experiment concluded that carbon dioxide is essential for the process of photosynthesis to take place. Now, can you explain how this conclusion could be drawn? So, negative starch indicates that the process of photosynthesis is absent in a leaf enclosed in the test tube. Now, जहाँ पे negative starch test हुई, it indicates that the process of photosynthesis didn't occur in a test tube which was, or in a leaf which was enclosed, or you can say in a leaf which was dipped in the test tube. This portion of leaf is getting all necessary requirements like light, water and chlorophyll except the carbon dioxide that is absorbed by KOH. So, the dipped leaf is getting all the requirements like chlorophyll, light, water. But the one thing it is lacking is carbon dioxide because that carbon dioxide is absorbed by KOH. Now, certain early experiments related to the process of photosynthesis. So, first is the experiment of Priestley. Joseph Priestley in from 1732 to 1804, in 1770 performed a series of experiments that revealed the essential role of air in the growth of green plants. So, Joseph Priestley in 1770 performed the experiments on the green plants and he showed the role of air in the growth of green plants. His experiment showed the role of the air in the growth of the green plants. Priestley discovered oxygen in 1774. He observed that the candle burning in a closed space, that is a bell jar, soon gets extinguished. इसका फिगर हम नेक्स्ट स्लाइड में देखेंगे. So what Priestley observed? He observed that when a burning candle was placed in a closed bell jar, यानी कि एक बेल जार था and यहाँ पे burning candle उसमें रखी. So this is a closed bell jar. Now यहाँ पे burning candle when it was kept within a very short period of time उसने ये देखा कि this candle it got extinguished. He observed that the candle burning in a closed bell jar it soon gets extinguished. Similarly, a mouse would soon suffocate in a closed space. Same way, यहाँ पे जब mouse को उसने रखा, that mouse also suffocated in a closed space. Okay? So he concluded that a burning candle or an animal that breathed the air both somehow damaged the air. उसने क्या conclude किया? पहले क्या observe किया? Burning candle when kept in a closed bell jar, it soon gets extinguished. Same way, when a mouse is kept in a closed space, it would soon suffocate. So what conclusion did he derive? उसने ये conclude किया कि burning candle या animal जो air को breathe करते हैं वो कोई ना कोई वे से air को damage कर रहे हैं. He concluded that a burning candle or an animal that breathe the air, जो air को breathe करते हैं, somehow both of them damage the air. But when he placed a mint plant in the same bell jar, he found that mouse stayed alive and the candle continued to burn. अभी ये बेल जार में जब उसने मिंट के प्लांट को रखा, so what did he observe? He observed that mice, they stayed alive as well as the candle continued to burn. देखो पहली एक्सपेरिमेंट है, ये क्लोज्ड बेल जार है कि जिसमें लिविंग माउस है एंड ये बर्निंग कैंडल है. आफ्टर सम टाइम द माउस सफोकेटेड एज वेल एज जो कैंडल बर्निंग थी वो क्या हुई, एक्सटिंग्विश हो गई. नाउ in a same setup he grew the mint plant. Now when he kept the mint plant in a bell jar and at that time he found that, देखो ये बर्निंग कैंडल है, ये माइस है एंड ये मिंट प्लांट है. सो ये टाइम पे कैंडल इट कंटिन्यूड टू बर्न, द माइस डिडंट सफोकेटेड, दैट इज इट स्टेड अलाइव. सो from this he gave one hypothesis. What hypothesis did he give? Plant restore to the air whatever breathing animals and burning candles remove. उसने क्या कंक्लूड किया कि बर्निंग कैंडल या एनिमल्स, ब्रीदिंग एनिमल्स जो चीज़ एयर में से निकाल रहे हैं, दैट मटीरियल इज रिस्टोर्ड बाय द प्लांट. ब्रीदिंग एनिमल, बर्निंग कैंडल, ये दोनों को क्या रिक्वायरमेंट है, ऑक्सीजन. ये ऑक्सीजन वो कहाँ से ले रहे हैं, एटमॉस्फेयर. ओके, प्लांट क्या कर रहे हैं, जो ऑक्सीजन एनिमल्स ने या बर्निंग कैंडल ने लिया वो फिर से कहाँ पे दे रहे हैं, एटमॉस्फेयर में. सो प्लांट रीस्टोर टू द एयर whatever breathing animals and burning candles remove. The next experiment is of the experiment of Jain INGENHOUZ. Jain INGENHOUZ from 1730 to 1799, he used a similar setup as the one used by Priestley. Priestley ने जो setup use किया था, same setup was used by Jain. But he placed it once in dark and once in sunlight. लेकिन उसने क्या किया, that setup he placed for one time in dark and then for one time in sunlight. It showed that sunlight is essential to the plant process. इसके ये सेटअप से उसने ये कंक्लूड किया कि प्लांट को वर्क करने के लिए सनलाइट इज वेरी मच एसेंशियल. It somehow purifies the air fouled by burning candles or breathing animals. See, burning candles, breathing animals ने एटमॉस्फेयर में क्या रिलीज किया होगा, कार्बन डाइऑक्साइड. अभी ये कार्बन डाइऑक्साइड एयर को डैमेज कर रही है. सो ग्रीन प्लांट्स क्या कर रहे हैं, ये डैमेज्ड एयर को प्योरीफाई कर रहे हैं. सो इट समहाउ प्योरीफाइज the air which is fouled by burning candles or breathing animals. INGENHOUZ is in an elegant experiment with an aquatic plant showed that in bright sunlight small bubbles were found around the green parts while in dark they did not. Okay, उसने एक दूसरा conclusion भी दिया. When he was working on the aquatic plant, he showed that in the bright sunlight, small bubbles were found around the green parts of the plant, whereas in the dark, these bubbles were not formed. Later, he identified these bubbles to be oxygen. बाद में उसने क्या conclude किया, ये जो small bubbles बन रहे थे, these bubbles were of oxygen. Hence, he showed that it is only the green parts of the plant that could release oxygen. See, उसने क्या देखा? Aquatic plant in the bright light, they showed the presence of the bubble near the green parts, whereas in the dark these bubbles were not formed. Later, when the bubbles were checked, it showed the presence of the oxygen. From this, he concluded that it is only the green parts of the plant which release the oxygen. We know that at the time of photosynthesis, oxygen is released. But who releases that oxygen, or from where that oxygen is released? So basically, oxygen is released only from the green parts of the plant. Now next is the experiment of Julius von Sachs. It was not until about 1854 that Julius von Sachs provided evidence for the production of the glucose when plants grow. So ये जो scientist था Julius von Sachs, he provided the evidence, he provided the proof that when the green plants grow, they did the production of the glucose. When the plants grow, there will be the formation or production of the glucose. Now, whatever glucose is formed, normally it is stored as starch. कभी भी glucose के form में नहीं होगा, it will be converted into starch. His later studies showed that the green substance in the plant, which is chlorophyll as we know it now, is located in special bodies which were later called chloroplast within the plant cell. Okay, उसकी studies से हमें ये भी मालूम पड़ा कि plant contain the green colored substance which is called as chlorophyll, and this chlorophyll is stored in the, or it is located in the special parts, in the special organelles which we know it as the chloroplast, and these chloroplasts are found within the plant cells. He found that green parts in the plant is where glucose is made, and that glucose is usually stored as starch. उसने ये भी observe किया कि it is the green parts of the plant which will prepare the glucose. Glucose will be prepared only in the green parts of the plant, and this glucose after synthesis is converted, or it is stored in the form of the starch. The next is the experiment of T.W. ENGELMANN in 1843 from 1909. So using a prism, he split the light into the spectral component. He took this prism, and using that prism, he split the light into the spectral components, into different spectral components. Then he illuminated a green alga cladophora placed in a suspension of aerobic bacteria. Now, इस प्रिज्म से उसने लाइट को स्प्लिट कर दी. Now, ये लाइट के कंपोनेंट से उसने किसको इल्यूमिनेट किया? ग्रीन algae, which was called as cladophora, and this cladophora was placed in the suspension of the aerobic bacteria. ये aerobic bacteria क्यों उसने यूज़ किए? The aerobic bacteria were used to detect the sites of the oxygen evolution. Aerobic bacteria थी, यानी they will need oxygen for their activity. So these aerobic bacteria basically were used to detect the sites for the production of the oxygen. He observed that the bacteria accumulated mainly in the region of blue and red light of the split spectrum. See, prism से उसने क्या किया? लाइट को स्प्लिट की. अभी ये स्प्लिट लाइट पे उसने किस पे एक्सपोज की? ग्रीन अलगा. ग्रीन अलगी कहाँ पे ग्रो कर रहे थे? इन द सस्पेंशन ऑफ द एरोबिक बैक्टीरिया. क्यों एरोबिक बैक्टीरिया को यूज़ किया? टू डिटेक्ट द साइट्स ऑफ द ऑक्सीजन इवोल्यूशन. सो उसने क्या कंक्लूड किया कि बैक्टीरिया they mainly accumulated in the region of blue and red light of the split spectrum. उसने ये ऑब्जर्व किया कि ये स्प्लिट वाइट लाइट स्प्लिट हो चुकी थी डिफरेंट कंपोनेंट्स में. सो जो कंपोनेंट बेसिकली जहाँ पे रेड एंड ब्लू लाइट जा रही थी, एट दैट रीजन द बैक्टीरिया वर accumulated. The bacteria accumulated in the region of which light? The blue and the red light. So a first action spectrum of photosynthesis was thus described. Now what is action spectrum? Action spectrum means it is a spectrum of the photosynthesis which shows the which wavelengths are used by the plants to create energy. Action spectrum, it is a spectrum for the photosynthesis जिससे हम we can know which wavelength of the lights will be used to create the energy. Which wavelengths of the light when absorbed will be used to generate the chemical energy. Okay. Now, ये जो action spectrum बना, it resembles roughly the absorption spectrum of chlorophyll A and chlorophyll B. Now, chlorophyll A and chlorophyll B are what? They are the molecules. They are the pigments which absorb the light. So, what is absorption spectrum? It is a spectrum which shows which wavelengths are most absorbed by the specific molecule. अभी जो भी pigments light को absorb कर रही है, okay. Say for example, chlorophyll A is a pigment, chlorophyll B is a pigment, xanthophyll is a pigment. So ये जो सब pigments है, वो कौन से wavelength की light absorb कर रही है, that is called as absorption pigment, and action spectrum that is called absorption spectra. And what is action spectra? Which wavelength of the light is used to create the energy. कौन सी wavelength light की energy generate कर सकती है, that is called as action spectrum. And a specific molecule absorbs which wavelength of the light, that is called as the what? It is called as the absorption spectrum. So जो action spectrum जो बना, it was resembling what? It was resembling the absorption spectrum. So that is the experiment. Now, how middle of 19th century is an important reference to photosynthesis. By the middle of 19th century, the key features of plant photosynthesis were known, namely that plant could use light energy to make carbohydrates from carbon dioxide and water. So, by the middle of 19th century, certain important features of process of photosynthesis were known. Like, क्या मालूम पड़ गया कि the plants they use the light energy to synthesize the carbohydrates from carbon dioxide and water. Plant light energy को use करके carbohydrates का production करते हैं, किसमें से? Carbon dioxide and water. The empirical equation representing the total process of photosynthesis for oxygen evolving organism was then understood as, okay, so oxygen evolving organisms कैसे photosynthesis करते हैं, या वो एक equation general equation दिया, using carbon dioxide and water in the presence of sunlight, the plant prepares CH2O and O2. CH2O is what? It is a carbohydrate. So, in the presence of, ये पूरा एक empirical, एक general form है photosynthesis का, कि plants they utilize carbon dioxide, water in the presence of what? Sunlight. And they prepare the carbohydrate and release the oxygen. So, CH2 here represents a carbohydrate which is mainly glucose, a 6-carbon sugar. Next is the contribution of Cornelius van Niel. A milestone contribution to understanding of photosynthesis was that made by microbiologist Cornelius van Niel in 1897 to 1985, who based on his studies on purple and green bacteria demonstrated that photosynthesis is essentially a light dependent reaction in which the hydrogen from a suitable oxidizable compound reduces carbon dioxide to carbohydrate. Okay. So Cornelius van Niel, उसने बहुत एक इम्पॉर्टेंट कंक्लूजन दिया He was basically studying on purple and green bacteria. While his study on purple and green bacteria, he demonstrated, he observed, he showed that photosynthesis is a light dependent reaction. That is, photosynthesis process occurs in the presence of light. In this reaction, a hydrogen from a suitable oxidizable compound, हाइड्रोजन कोई एक ऑक्सीडाइजेबल कंपाउंड में से रिलीज होकर कार्बन डाइऑक्साइड को कार्बोहाइड्रेट में रिड्यूस करेगा. व्हाट डिड ही शोड? ही शोड दैट हाइड्रोजन फ्रॉम अ ऑक्सीडाइजेबल कंपाउंड इज रिड्यूसिंग द कार्बन डाइऑक्साइड टू कार्बोहाइड्रेट. ये पर्टिकुलर स्टेटमेंट को हम एक इक्वेशन के फॉर्म में कैसे डाल सकते हैं? सो दिस इज अ ऑक्सीडाइजेबल कंपाउंड एंड दिस इज अ कार्बन डाइऑक्साइड. सो व्हाट डिड ही सेड? अ हाइड्रोजन फ्रॉम अ ऑक्सीडाइजेबल कंपाउंड, इसमें से हाइड्रोजन रिलीज होगा. ये हाइड्रोजन को एक्सेप्ट कौन करेगा? ये कार्बन डाइऑक्साइड. ये प्रोसेस किसके प्रेजेंस में होती है? लाइट के प्रेजेंस में. सो ये जो ऑक्सीडाइजेबल कंपाउंड है उसका हाइड्रोजन कार्बन डाइऑक्साइड ले लेगा एंड ये कार्बन डाइऑक्साइड रिड्यूस होकर क्या बनाएगा? इट विल फॉर्म द कार्बोहाइड्रेट. अभी ये जो ऑक्सीडाइजेबल सब्सटेंस था उसमें से हाइड्रोजन निकल गया. सो दिस इज वो खुद ऑक्सीडाइज हो गया एंड अ मॉलिक्यूल ऑफ वाटर विल बी रिलीज. So this is a equation form of Cornelius von Neal experiment. उसने ये दिखाया कि light is essential for the process of photosynthesis to occur where hydrogen from a oxidizable compound reduces carbon dioxide to carbohydrate. ये इक्वेशन में फिर से समझाती हूँ. देखो, दिस इज ऑक्सीडाइजेबल सब्सटेंस के जिसमें हाइड्रोजन है. ये हाइड्रोजन इसमें से निकलकर कौन एक्सेप्ट करता है? कार्बन डाइऑक्साइड. सो दिस कार्बन डाइऑक्साइड इज कन्वर्टेड टू अ कार्बोहाइड्रेट. इन ग्रीन प्लांट्स वाटर इज द हाइड्रोजन डोनर एंड इट इज ऑक्सीडाइज्ड टू ऑक्सीजन. हमने क्या बोला? हाइड्रोजन फ्रॉम अ ऑक्सीडाइजेबल सब्सटेंस. सो ग्रीन प्लांट्स में वो ऑक्सीडाइजेबल सब्सटेंस कौन सा है? वो कौन सा सब्सटेंस है जो हाइड्रोजन को डोनेट करता है? सो इन ग्रीन प्लांट्स द हाइड्रोजन डोनर इज वाटर. वाटर में से H2 निकल जाएगा, यानी वाटर का क्या हो गया? जब कोई भी सब्सटेंस में से हाइड्रोजन का रिमूवल होता है तो वो सब्सटेंस क्या होता है? ऑक्सीडाइज होता है. सो इन ग्रीन प्लांट्स द डोनर ऑफ हाइड्रोजन इज वाटर. वाटर में से हाइड्रोजन रिमूव होगा, वो खुद ऑक्सीडाइज होकर किस में कन्वर्ट हो जाएगा? ऑक्सीजन में. Now, there are some organisms which do not release oxygen during photosynthesis. There are certain few organisms which never releases oxygen at the time of photosynthesis. So that photosynthesis where photosynthesis के process तो हो रही है but oxygen release नहीं हो रहा है. So that photosynthesis is called non-oxygenic and anaerobic photosynthesis. So we will take the example of anaerobic photosynthesis के जहाँ पे oxygen release नहीं हो रहा है. When H2S instead is the hydrogen donor for purple and green bacteria, the oxidation product is sulfur or sulfate depending on the organism and not oxygen. Okay, जब water hydrogen donor है तब what is the product? Oxygen मिलता है. But when instead of water H2S, that is hydrogen sulfide, is used as the hydrogen donor, this happens in the case of blue and green sulfur bacteria. कि जहाँ पे इंस्टेड ऑफ वाटर H2S is used as the hydrogen donor. In such cases, the oxidation product is not water. See, दो कंडीशन है. जब water is hydrogen donor, the oxidation product is oxygen. But when H2S is the hydrogen donor, here the product is either sulfate or it may be sulfur also. Okay. So, in the purple and green sulfur bacteria, hydrogen donor is not water, it is H2S. So, instead of water, here the product will be sulfur or sulfate. So, such photosynthesis is a reaction where oxygen is not released. So, he inferred that oxygen evolved by the green plants come from water and not from carbon dioxide. अभी क्वेश्चन होना चाहिए कि वाटर एंड कार्बोहाइड्रेट ये दोनों यूज़ हो रहे हैं. तो हमने ये कंक्लूड तो कर दिया कि ग्रीन प्लांट में ऑक्सीजन जो आता है वो वाटर में से आता है एंड ना के CO2 में से. अगर CO2 में से आता तो पर्पल और ग्रीन बैक्टीरिया, पर्पल एंड ग्रीन सल्फर बैक्टीरिया को भी ऑक्सीजन रिलीज करना चाहिए, बट यहाँ पे तो ये हुआ नहीं. पर्पल एंड ग्रीन सल्फर बैक्टीरिया did not release the oxygen. So from this he gave a very important conclusion कि जो plants oxygen को evolve करते हैं, that oxygen comes from water and not from the carbon dioxide. This was later proved using the radioisotope techniques. So, the correct equation that would present the overall process of photosynthesis is, so the general equation for the photosynthesis is 6 CO2 plus 12 water give in the presence of light gives C6H12O6, which is glucose, plus 6 molecules of water plus 6 molecules of oxygen. This is the equation of the photosynthesis. The oxygen released is from water. जो oxygen release हो रहा है, that comes from the water. This was proved using the radioisotope techniques. Note that this is not a single reaction but description of multi-step process called photosynthesis. अभी ये जो reaction है ये single reaction नहीं है. It is a multi-step process which is called as the photosynthesis. So, how can we say what is photosynthesis? Photosynthesis is a collection of oxidation reduction reaction, or you can say it is a redox reaction. It is a redox reaction. Redox reaction के जहाँ पे oxidation reduction होता है. Oxidation यानी water is oxidized to oxygen क्योंकि वहाँ पे loss of electrons, or you can say there is the loss of hydrogen atoms. Whereas reduction, where carbon dioxide is reduced to carbohydrate, that is gain of electrons. In some bacteria, oxygen is not evolved and is called non-oxygenic and anaerobic photosynthesis. The examples are green sulfur, purple sulfur and green filamentous bacteria. These are the organisms के जहाँ पे oxygen is not released as at the end of the photosynthesis क्योंकि यहाँ पे hydrogen donor H2S है. It is not the water, it is the H2S, and as H2S is used, it won't release the oxygen. Now हमने एक ये reaction देखा. What was the reaction of photosynthesis? 6 CO2 plus 12 water gives C6H12O6 plus 6 H2O and 6 molecules of oxygen. So यहाँ पे एक question आता है कि why 12 molecules of water as substrate are used in the equation. जो photosynthesis का equation था उसमें क्यों 12 water molecules हुए हैं. ये अभी explanation थोड़ा आपको difficult लगेगा, but still I will explain you. When you study the next part, or when you study the बायोसिंथेटिक फेज ऑफ फोटोसिंथेसिस, ये टाइम पे आपको ये टॉपिक ज़्यादा क्लियर होगा. बट फॉर योर एक्सप्लेनेशन, फॉर योर नॉलेज, little I will share. Products of light reaction required in dark reaction to form one molecule of glucose are 12 molecules of NADPH and 18 molecules of ATP. Now, थोड़ा समझाती हूँ कि फोटोसिंथेसिस 2 स्टेजेस में होता है. वन इज़ लाइट फेज एंड डार्क फेज. डार्क फेज पे टाइम पे ग्लूकोज बनता है. सो नाउ स्टूडेंट्स, ये डार्क रिएक्शन है, बट डार्क रिएक्शन के टाइम पे लाइट रिएक्शंस, जो रिएक्शंस लाइट के प्रेजेंस में होते हैं. सो एट द टाइम ऑफ डार्क रिएक्शंस द प्रोडक्ट ऑफ द लाइट रिएक्शंस आर रिक्वायर्ड. So which products of the light reactions are required at the time of dark reaction? So they are 1 molecule of the glucose formation. To form 1 molecule of glucose, there is a need of 12 molecules of NADPH, nicotine amide diphosphate. Okay. So 12 molecules of NADPH and 18 molecules of ATP are required to form 1 molecule of glucose during the dark process. But ये NADPH and ATP कब बनता है? लाइट रिएक्शन. उसका यूज डार्क रिएक्शन में होता है, बट प्रोडक्ट्स बनते कब है? लाइट रिएक्शन में. For forming 2 molecules of NADPH from NADP plus, NADPH किस में से बनते हैं? NADP plus में से. So to form the 2 molecules of NADPH from NADP plus, there is a requirement of 2 molecules of water which will demand 4 electrons and protons can be satisfied. So for forming 2 molecules of NADPH from NADP plus, 2 water molecules are required so that the demand of 4 electrons and protons can be satisfied. ये इक्वेशन अगर एक्सप्लेन करना है, 2 water and 1 NADP plus will give 2 NADPH plus 2 protons, that is 2H plus, and oxygen will be released. The 2 molecules of water split to form 1 molecule of oxygen, and the respective number of electrons and hydrogen ions are formed as shown in the reaction below. 2 molecules of water will split and form 1 molecule of oxygen, and the respective number of the protons and electrons are formed. See students, नीचे के reaction में आप देख सकते हैं कि 2 molecules of water split in the presence of light, which forms 4 electrons plus 4 protons and the molecule of oxygen. So students, so for producing 12 molecules of NADPH, 12 molecules of water will be required. To form 12 molecules of NADPH, how many molecules of water will be required? 12 molecules of the water are required. So that is regarding the history of the photosynthesis. I end my lecture here. In the next lecture, we will be studying where the process of photosynthesis occur and how the process of photosynthesis will take place. I end my lecture here. Thank you students.